Hello and welcome to the GCSE Biology tutorial for topic B6 Inheritance, Variation and Evolution. This is part 3 and will cover variation. We'll also be looking at selective breeding, genetic engineering and cloning. Starting off with, what is variation? Well, you'll need to know that there's two types. Genetic variation, which is caused by the genes, so they're passed on from parents to offspring, and every time a new offspring is created, a new combination of genes from the parents is brought together. So no two offspring will be genetically identical, apart from identical twins, that is. An example of genetic variation would be things like eye colour, blood group, inherited diseases. Another example of variation is environmental variation. Now, this is caused by the environment and the conditions that an organism needs to live and grow. So for a plant, that could be water or sunlight. For humans, that could be the food that they eat. There's a lot of variation within humans because of the environment. But most characteristics are actually due to a combination. So for example, someone's weight is down to genetics and the environment. So one way we can use variation to help us is selective breeding. Now, selective breeding is quite literally what it says, selecting and then breeding. So it's when farmers or gardeners mainly select an animal or plant that they want and then another to breed together, and they would choose desirable characteristics. So they would choose certain factors that they want. For example, in a cow, they might choose features such as having more meat or producing more milk or being more fertile. In plants, it could be crops that are more disease resistant or bigger or riper. By choosing these organisms and breeding them together over many, many generations, you get the desirable characteristic developing over and over again. And of course, every time these animals mate and produce new young, they're not all going to have that desirable characteristic. So the whole key with selective breeding is it continues for many generations. You keep breeding and breeding again. Now the good thing about this is it's been going on for years, decades in farming, without even understanding the term genes. And it's allowed for desirable characteristics to be selected. However, by constantly inbreeding, so breeding offspring with offspring, it reduces the gene pool, so there's less genetic variation. And that's a problem because if there was, for example, a disease that came in to a stock of animals that were all very similar genetically, then if one wouldn't be able to combat the disease, it would be more probable that others wouldn't either. It also means that if a genetic disease or genetic defect was bred by accident, that would be passed on. So there is issues with selective breeding. A more recent technology is genetic engineering. Now, this involves transferring or replacing or modifying a gene for a desired characteristic from one organism and then maybe placing that into another organism. For example, you can take the genes to make insulin and put them in bacteria to make insulin for diabetics. GM stands for genetically modified, so GM crops you can modify so they're bigger or they can grow where there's disease or drought. And as we've mentioned before in the previous topics, we can use genetic engineering in gene therapy. So that's not to cure genetic disorders, but to help treat them. Now, an example that's useful to know is how insulin is produced in genetic engineering. So let's just go through this diagram here. You have a human cell and you will take the gene out for the gene that codes for making insulin. So they would isolate the chromosome and then cut the gene out using enzymes, and that gene codes for making insulin. You will then insert that gene into a vector. Now, a vector is something that's going to carry that gene. Bacteria are very good at being vectors because they multiply so quickly. The bacteria has a plasmid, this circular loop of DNA, and that's cut open, and the gene from the human cell for insulin is inserted with an enzyme and sealed. The bacterium is then left to reproduce, and as it reproduces more and more of the gene that's inside it, inside its plasmid. So it produces more and more of this insulin. And then using biotechnology, which we will look at in other topics, you can then kill the bacteria and be left with the insulin. So that's an example where you can use a vector, but you can also take the gene itself and put the vector into another organism. So then the gene is inserted into new cells. And a good point to do that at is when it's an embryo, because those cells haven't specialised yet. So if you insert the genes then into the embryo cells, then all the cells will start to copy and multiply and be the same. Now, the good thing about genetic engineering is, of course, it increases the yield where there's food shortages, make sure there's lots of food available. Gene therapy we mentioned before, so treating cystic fibrosis, for example. But you can also modify plants to make them even better. So you could put extra nutrients in them, like antibiotics, or another example is golden rice, and that has been genetically modified to contain beta carotene, and that helps people that don't have enough of this nutrient. And if they don't have enough of it, it can cause blindness. So this rice is like a special rice with extra nutrients in it. Now the issues are, if you start to affect the amount of organisms in a particular area, it could affect the other organisms that already live there. So it could affect food webs. So if one population of food goes up, another could go down, and it could result in some species actually being reduced. So it could reduce biodiversity. There is also concern about allergies from GM food, so side effects. The thought of creating plants that then you couldn't destroy, so superweeds, and cross-pollination, so the genetically modified pollen or eggs from one plant getting into another, not of the same type and not the one that you're wanting to change. At the moment, genetic engineering with plants is carried out in controlled situations, but there is these concerns. Another process you'll need to know about is cloning. There's a couple of different types of cloning. The first one we're going to talk about is adult cell cloning. So this is the most famous example you might know is Dolly the sheep. So Dolly the sheep was a clone, but how was she actually made? So you need to listen really carefully to how it happens, because there's quite a few different steps involved. So first of all, you would take an unfertilised egg cell from a female sheep, and you remove the nucleus. Okay, so you don't need that nucleus anymore, you remove it. Then you would take another sheep, and this is the sheep that you want to clone, and you would take an adult body cell from that sheep, so anything from the skin or the body itself, something with the full set of chromosomes, and you take the nucleus out of that body cell and put it into the empty egg cell that you just created. Now, because they're not going to join naturally, you'd have to give an electric shock, so it's stimulated to form an embryo. And then when the embryo is large enough and a ball of cells, it can be implanted into another sheep, which will just carry it in its uterus, so it's the surrogate. And once the embryo grows and the baby sheep is born, it will be a genetically identical clone to the original sheep that you took the body cell from. So it won't be a clone to the surrogate, it won't be a clone to the one that you took the egg cell from, it will be a clone from the one you took the body cell from. Now the good thing about this, you can produce genetical copies, it increases studies of embryos and ageing, and actually it can help us preserve endangered species. But again, the issue is reducing that gene pool. If you have less variation, there is more chance that diseases could cause harm. Dolly the sheep actually died early, and she had arthritis, so some people are concerned there was issues with growth in cloning, and also unethical concerns. At the moment, there's no cloning of humans, but what people are concerned that cloning could be taken out of hand and go too far. Another type of cloning is tissue culture, which is using plants. That's when cells are taken from a plant, or groups of cells called tissues, and then grown in the special agar or nutrients and given auxin, that plant hormone, to help it grow. And they make tiny little plantlets which can then be put into compost and grown. So you quickly get lots of plants from one plant. The good thing about this is you can make it in small areas, increases the yield, and they grow quickly. However, again, it is decreasing that gene pool. So when I say gene pool, I mean all the genetic variation is making that less of that, which increases the chance of, say, for example, a plant getting a disease. They could all get the disease if they were all the same. So here's some questions. If you pause the tutorial and attempt these now. Okay, so the two types of variation are genetic, like eye colour, and environmental, like body weight. Given a concern of selective breeding, it can reduce the gene pool and increase chance of genetic defects. Give one useful way that plants could be genetically modified. Well, they can be made bigger, there's more plants, so bigger yield, resistant to diseases, extra nutrients like golden rice. What is tissue culture? That's where cells are taken from a plant, a group of cells, which is called a tissue, grown in agar and auxin to produce little plantlets, which are then grown in compost and are genetically identical to the original plant that you took it from. There is another kind of ways which you can take and grow plants by cloning, which is not tissue culture, and that is called cuttings. And that's just when you take a cutting of a plant and then grow it. But it's why it's not called tissue culture is you're not taking like a group of cells, you're taking an actual big section of the plant off. Okay, so 6-mark question is describe how Dolly the sheep was created. So for this one, you need to talk about adult cell cloning. Okay, so this is when the body cell is taken from the original sheep and the nucleus is removed, and then you take the nucleus take that nucleus and put it into an that bit again. So you take a body cell from the sheep you want to clone and you take the nucleus out of that. You put that nucleus into an empty, unfertilised egg cell. You then give it electric shock therapy to turn it into an embryo. And when it's grown into a ball of cells, you can put that into a surrogate. And when the sheep gives birth, the offspring will be a identical clone to the original sheep you took the body cell from. So in theory, you could have 3 sheeps involved here. One that's providing the egg cell, which you're going to take the nucleus and discard. One that is providing the body cell, which you're going to use the nucleus from. And thirdly, the surrogate sheep. And this diagram just reinforces what we've mentioned already. And there's nothing wrong with adding diagrams and questions as well. In fact, that will help the examiner understand your detailed as this, but they might be needed just to explain the cloning process. So in summary, we've looked at variation, selective breeding, genetic engineering and cloning. Thank you.